tenemos fechas duras de límites porque queremos que se abra el año que viene. Este año nos vimos en la necesidad penosa de no abrir ese magíster, porque como lo estábamos cambiando era engañoso decirle a un chico, entre un magíster que vamos a cerrar el año que viene. Por acá tenemos a Marlon, a Brandon, que diga que está de los más avanzados en ese programa y espero que termine pronto. Yo sé que ha entregado tesis y demás. Entonces, bueno, esa es la primera información. La segunda información es referente al otro magíster que está coordinando el doctor Casanova, que también está casi listo, que hemos tenido algunos inconvenientes ahí técnicos, pero creo que estamos listos para lanzarlo este año también, máximo el año que viene, que es un magíster profesional en medicina y cirugía de animales, que también está reformado, porque antes era, ¿cómo se llamaba antes, Tomás? En
pequeños animales.
De pequeños animales. Ahora no queremos que sea solo de pequeños, sino que incluya grandes, exóticos y mascotas. Entonces es un magíster profesionalizante que también es muy útil porque tenemos muchos colegas que no están metidos en el sistema académico de progresión como asistentes, titulares y demás, sino que son colaboradores o plantas de la clínica y les cuesta mucho trabajo poder avanzar porque entrar a un magíster académico es más complicado. Pero este magíster, además de resolver eso, resuelve problemas reales de la medicina veterinaria local. Entonces creemos que podemos fortalecer harto el programa de posgrado con el doctorado consolidado y con la disrupción de estos dos magísteres. Ojalá que los dos se vayan a ver. El último que conversé estuvo a punto de salir el año pasado, el antes pasado, pero hubo algunos inconvenientes técnicos ahí con la planta académica que ya están resueltos en su mayoría. Entonces eso era lo que yo quería traerles en términos de lo que ha pasado. Este año terminamos con 30 graduados ya del doctorado, lo cual, si uno se pone a pensar, hemos generado empleo para 30 personas porque todos están en la academia o en algo parecido. O sea, de alguna manera es una carrera también, o sea, que da empleabilidad y que le da futuro a las personas. Estamos muy felices con eso. Y estoy feliz de que estén acá estudiantes de los primeros años, de años un poco más adelantados, de los años terminales, José Luis, Ivana, y los chicos que están acá, Valentina, Marlon y Cata, que son más nuevos, y ahí supongo que nos estén escuchando Sofía, Marlene e Iván. Esto es lo que yo quería traerles, no tengo muchas más noticias que darles, solo pedirles que sigan siendo tan proactivos como hasta ahora, que sean interactivos con nosotros, que siempre estamos abiertos a recibir críticas para mejorar el funcionamiento del programa, que nos disculpen si a veces nos equivocamos y las cosas no salen como ustedes quieren, y a veces andamos cortos de recursos, nos piden para ir aquí o allá, y no siempre podemos apoyarlo, pero estamos tratando de siempre apoyar en todo. Agradezco que estén acá los estudiantes, agradezco a los académicos que hayan venido, porque esto es una actividad de una connotación especial desde el punto de vista científico, así que muy feliz de que estén acá. Ya, no quiero seguir, ahora voy a introducir al profesor que Mutien. Esto lo voy a hacer en inglés para que lo entienda y para que, bueno, quede como tiene que ser en inglés. So, it's a big pleasure for us as a program of postgraduate program to receive Professor Mutien Galigliani, who kindly accepted to give this talk today. Mutien, as I mentioned, has been very proactive and very keen with us, with the University of Concepcion. He has a kind of a symbolic position here as visiting professor of this university too. And if you don't know, he has accepted 3 PhD students from this university. One is sitting over there, Tomás Casanova, who finished the PhD with him. Then Jose Felipe Rivas, who is already finishing, I guess you can mention that, but he did already his research, he's finished the paper. And Sofia Rogliano, the one that I mentioned, is starting the PhD there. So Mutien is a worldwide expert in emerging diseases, not only in that, in airborne diseases, in arthropod and biology, and the different vectors that are in these animals. And he will share with us his experience in whatever he wants to talk about. He's always welcome. Thank you, Mutien, for coming, and the floor is yours.
Thank you, Fidel, for this introduction. And thank you, Thomas, for allowing me to present to you briefly what we do. So the aim of today's presentation is to show you what us, I would just say, simple vets can do in some very important questions for the emerging infectious disease. So my name is Lucien Garigliani. I'm a pathologist, and as you know, pathologists, they work mostly from the necropsy room and from samples they receive from clinicians. And by doing so, they are really in the first line when some emerging infectious disease is appearing somewhere, because we are facing the first cases of these animals which die from the infection, or the samples that the clinicians took from the sick and dead animals. Based on that, we are at the forefront of development of diagnostic tool to recognize, identify this emerging infectious disease. And something that I did during my PhD and I continue to do afterwards is also to develop the expertise to isolate these new agents and develop models. And these are quite simple and old methods, but they are still important. I will show you some examples of a mix of really well-known classical virological methods which are used since decades, and some very up-to-date new technologies, and both actually are complementary. The new techniques do not completely replace these old ones, so you will see that. Well, so as I told you, the pathologist has really a central role in medicine, in human and veterinary medicine, and this is what is shown here. It's very important, of course, for the diagnostic of disease, so as a help for clinician, important for research, of course, also, like reading histoslides, performing morphological diagnosis, but they are really at the forefront of the evolution of veterinary medicine. This is an important role of pathologists. And of course, this is implying the use of really extremely rapidly evolving methods. There are plenty of very exciting methods. I don't know if you guys know about this spatial omics that we are now using a lot in medicine, human and vet medicine. So all these techniques actually use histology as a basis, which is superimposed with data about the expression pattern at the level of proteins, which is called spatial proteomics, or RNA, which is called spatial transcriptomics. This allows you, for instance, to see how a given virus is able to enter the brain through which structures, and it's also explained, of course, the clinical signs that the patient will develop upon infection, for instance. And this is something very exciting. This is really recent, and we have the chance in our faculty to have access to such specific equipment. So this is our faculty that some of you know well, like Fidel, for instance, knows the faculty very well, and Thomas, of course. This is the vet faculty with the clinics here. My lab is there. Here there is a building which is interesting because we have new biosafety level 3 facilities here for the whole institution. And which is very nice there is that we have the vet faculty here, and about 500 meters in that direction we have the human hospital. So the faculty of medicine and veterinary medicine are really close to each other. And I think this geographical proximity is something very important. It explains how easily we are used now to work with each other, and how complementary, again, we are when facing, for instance, emerging infectious disease. A few words about Belgium, because many of you probably do not know Belgium. So Belgium is a very tiny spot here in Europe, so it's a very small country compared to Chile, of course. But it's a quite important country because it's you have the headquarters of NATO, and all the European institutions are centered there. So it's one of the countries with the highest population density in the world. So, but in spite of that, there are beautiful cities there, like Bruges, probably you know, Ghent. This is Dinan, very close to where I live. It's also beautiful. But we also have some nice landscape, of course, not like Chile. We cannot compare. I will not argue about that, and of course, Chile is wonderful. But we have some things that you do not have, guys. We are the inventors of fries. The Belgian waffles, chocolate, of course. You know, Tintin, Los Pitufos, or cervecitas también. Muchas cervecitas in Belgium. So, yeah. So, yeah, you want to visit Belgium when you see that. And we have a long history of collaboration with Chile, notably through an agency which is called Wallonie-Bruxelles International, which is actually financing a lot of exchanges programs between our countries. It's very interesting if you want to perform a short stay in a university in Belgium. This is a very, very nice way to apply for a small grant allowing you to travel and visit another lab. Yeah. And this is the city of Liège, where my university lies. That, again, some of you know very well. This is the train station. This is the human hospital. So, again, the vet faculty is there and the human hospital very close by. And this is the campus, the main campus. So it's quite nice because it's outside the city. So it's rather in the woods. It's beautiful. We also, as Fidel told, had worked hard on developing this joint supervision PhD program. I think it's very nice for a very good opportunity for PhD students to have the co-diplomation. I think it's very a good way to continue the collaborations. Now we'll give you a series of examples on how us vets with some expertise, like in my case in infectious disease, we can bring this expertise to very important questions in different disciplines, and also how the collaboration with disciplines is important. Well, this is a case from the faculty here, actually, with colleagues from Chile. So I'm very interested in the evolution of viruses, because when you sequence viruses, you generate sequence data, and you like to analyze them and perform phylogenetic analysis to see the evolution over time. And this might be quite important for very practical questions like the adequacy between the vaccines you have in your country to protect the animals and the strains of the virus which are circulating on the field. And sometimes you have vaccines which are not efficient in bringing protection to these strains. So this is why it's very important to update regularly these vaccines based on sequence data, strong sequence data, and this is what we did here in Chile. This is another example. This is something I'm quite proud of what we did in our university, is that during the pandemics, you know how difficult times we had in the human hospitals with a lot of patients dying from COVID-19 and with the lack of very efficient antiviral approaches. So what we did is that at the very start of the epidemics, the clinicians from the medical doctors from the hospital asked to the colleagues in the university who could help in isolating viral strains and developing tools to study the disease. And since we have BSL-3 facility in the vet faculty, we said, okay, we can do that. We send us the samples, we will isolate the virus, develop the tools. And quickly they said, okay, one thing we would need is that you guys qualify the plasma taken from convalescent patients. So people which got infected by the virus produced high amounts of protecting antibodies. And they said if you take this plasma, which is protecting against the virus, and you inject it to a severely affected patient admitted to the hospital, maybe you can raise the survival rate of this patient. Of course, this should be done very quickly upon admission at the time when the virus is still multiplying into the lung tissue, because you know that over time, most of the lesions you have in the lungs of these people are due to the overreaction of the inflammatory response rather than to the viral replication itself. Well, this is what we did. So we qualified thousands and thousands of plasma from these patients, and they applied this protocol. And one can see that actually by doing so, they decreased the mortality rate by 30% in these people. And this is something to me very interesting for, I guess, us veterinarians to be able to help the medical disciplines in this way. Also about what we do in digital pathology. So most publications, I would say high impact publications, require now that we do not just perform a qualitative evaluation of the lesions under the microscope, but we need more quantitative approaches using artificial intelligence based softwares which actually have an unbiased calculation of the lesions you want to highlight. And this is something very important, and so we are working hard now to develop these tools. We're doing more and more bioinformatics, if you like, and this is very important, a very important part of our work in my department. Another examples here, so you can superimpose different stainings or sometimes immunohistochemistry or immunofluorescence image and bright field image, and see how these stainings and lesions can co-localize in the tissue, and then you can interpret what is going on in the process you want to study. I think this is something very interesting. It's most of these tools, I'm saying that for pathologists, but they are free, so there are plenty of free softwares on the web. But the thing is that they are quite difficult to use. We need some training to get used to that, because it's just command line softwares. It's not so user-friendly, but you can do wonderful things. So this is very important of the evolution of pathology at the moment. Okay, also it's here another example. We do a lot of cooperation internationally, mostly in Southeast Asia, in Africa, and also a bit in South America. And for instance, in Southeast Asia, you know how important is the production of fish meat for the human consumption and for the economy of some countries like Vietnam, for instance. It's something very important there. And what they are facing now in Vietnam is, because of the climate change, the global warming, the salinity in the lower part of the Mekong Delta is increasing. And so now the salt concentration is so high that the breeding and the farming of these fish is becoming more and more difficult, because these fish just die because salinity is too high. And so what we decided to do here, it's a project led by genetician colleagues from the faculty, is that first we sequence the full genome of this fish species, because we need a genetic basis to do some genetic selection for fish individuals that were more resistant to salinity. So this is the first part. We sequenced the genome, and then actually based on genetic selection we were able to identify specific breeders which were much more resistant to salinity. And this is quite simple product, but it brings immediate answers to critical questions, because in the lower part of this delta it's mostly where most of the poorer farmers that live. So it's something very important for their survival and subsistence in the future. So this is an example of what we do. Yeah, now I will spend a bit more time on my main research focus, which, as Fidel said, is on arbovirus, so viruses that are transmitted by arthropods, like mosquitoes, for instance, and mostly on the host pathogen interactions. And these viruses are really becoming more and more important. I will also explain the situation briefly in South America, because they are emerging in most continents, and in Europe, of course, but also here in the Americas. In this genus Orthoflavivirus, you find plenty of known very important pathogens like Japanese encephalitis, West Nile virus, Zika, dengue, yellow fever, all very important pathogens. And one maybe that you don't know yet is a virus called Uzutu, which is closely related, as you see, to Japanese encephalitis and to a less extent to West Nile, and which is emerging everywhere and very recently emerged in South America. So I think it's important that you hear about that guy. And there are some also clinical clinical importance for people, because the condition in South America seems to be different from what was observed in other continents. I will explain you that in a few minutes. So this is a distribution of these viruses, like you see Zika. It seems to be also present in Chile, by the way. West Nile, yellow fever, well, plenty of important viruses. We have a lot of tick-borne encephalitis now. This is quite old, but now it's really present also in Belgium, this tick-borne virus. So what about this Uzutu virus? It all started with such impressive images where you had thousands and thousands of dead birds, mostly around the roads. They were hit by a car, actually. And why did it happen is that because they had some neurological conditions, so they were suffering from viral encephalitis. And it happened in 2001 in Austria. And so it was identified as the Uzutu virus, a virus which had been described in Africa a couple of decades before. And some retrospective analysis showed that it was already present since a couple of years in Italy. Okay, and this virus, like many of these viruses from the orthoflavivirus genus, they are transmitted by mosquitoes, and they have a wild reservoir, which actually is made of birds, so wild birds. And this is the endemic cycle, allowing the virus to spread or be present in a given region. But sometimes there is a spillover through the mosquito through other species like people or some animal species. Okay, and most of these infections in people are asymptomatic, but again, this is something which is evolving over time. I will show you that. So there were plenty of cases in Europe where people got sick because of this Usutu virus infection. It's a bit like West Nile, so it's mostly a flu-like symptom, so fever, but also neurological impairment, so sometimes very important neurological signs. A few people died from the infection as well, but mostly immunocompromised persons. But anyway, it can be a lethal infection. And how did it happen to emerge in Europe? This is one of the examples of how migratory bird can be important in the spread of viruses, because it was first described in the 1950s in South Africa, and then through migratory birds it spread northward to Europe, to the western and eastern migratory flyways here in Europe. So it was really brought by this migratory bird. It's very similar to influenza virus H5N1 that you probably know, which is also has been spread globally because of these birds. So we analyzed actually what happened, and this is what we called in genetics a bottleneck, where you have this main introduction event there of a strain of the virus, and then the virus actually adapted itself to the local bird species and mosquito species that are present in Europe and not present in Africa. So there was a selection for the best adapted variants of this virus to better replicate in this host, so the mosquito bird cycle of transmission. And now it's quite stable and has become totally endemic in Europe, in most of Europe. These are the flyways in Africa, Europe, and the Americas that you probably know about. Yeah, this is the presence of Usutu virus and West Nile virus. And you can see that in many countries they actually are circulating. And why is it important? It's because actually in the areas where there is a high endemicity for both viruses, like the north of Italy, there you have Venice, so here is the delta of the Po River. And this is a very important place for migratory birds which come from Africa, stop here before they go, for instance, to Scandinavia during the summertime. So there are a lot of these migratory birds which stop in that place. And this is where it's like a hotspot for emerging infectious disease. And there actually you have plenty of West Nile and Usutu. And what they observed in people there in that region is that most people are seropositive for Usutu antibodies. So they have been facing this virus. And very interestingly, since Usutu is less virulent than its West Nile cousin, it's actually acting as a vaccine. So people which are seropositive against Usutu actually are protected against a severe disease after a West Nile virus infection. This is very nice because there is no vaccine against West Nile so far in people. There are some in horses, for instance, but there are no vaccines in people. So this is a very interesting case of the competition between two closely related viruses for the same ecological niche, and how finally the emergence of this virus is rather good news for human and public health because it's protecting the people. Okay. So this is also the basis probably of future vaccines against West Nile based on attenuated strains of Usutu. So we have colleagues here in Italy which hardly work on that, and that's very interesting for the future. A bit like the vaccines against yellow fever is also an attenuated strain of yellow fever virus, for instance, widely known. So what about Latin America? There are plenty of such papers saying that it's the next emerging threat for Latin America, and until recently I thought it was not present yet on the continent, but actually This is a recent paper here in Colombia. And here, it's very small, but I can read it. They found a quite high seroprevalence against Usutu virus in patients which showed a dengue-like syndrome. So it's a bit different from what we are experiencing in Europe, where it's mostly a neurological condition with signs of encephalitis. But here it's more like yellow fever with jaundice, flu-like symptoms, of course, hemorrhagic syndromes. So more like dengue, yellow fever. It's quite different, well, totally different from what we observe in Europe. It had never been described before, this clinical syndrome. And they had, for instance, 9 patients that they described here with Usutu positive patient with really severe clinical condition. This is unusual. So it means that in the differential diagnosis of this dengue-like syndrome, Usutu virus become something very important. We do not know how this specific Usutu strain did emerge there in Colombia, because it's really in the middle of Colombia. I have no clue about that, but it probably will spread quickly across the continent. I think this is something very important here. And as you understood, it's affecting birds. You can have mass mortalities in birds, but of course also here people. It's a very nice paper to read. Okay, what about birds? Because we are vets, so of course we are also interested in what is going on in animals. What we know, you have seen all the dead birds that I showed you a bit earlier were what we call blackbirds, so Turdus merula. But also night raptors are highly susceptible to infection. And all passeriforms and strigiforms are all extremely susceptible to the infection by Usutu, while plenty of other bird species are exceedingly resistant, like domestic chicken, geese, and so on. Ducks are extremely resistant. We do not know why. And we had that question, and this is the PhD actually from Felipe Rivas. And we asked ourselves, okay, what could underlie such very different phenotypes in these bird species? So the question is, what are the viral and host factors involved in this difference in terms of susceptibility in avian species? We developed models, in vivo models, in the canary, for instance, here. And you can see that upon infection in the skin, so we use a needle and a syringe to inject intradermally the virus here, a bit like the mosquito is doing during the natural infection. While feeding on the blood here, it's also injecting the virus in the epidermis and dermis and creating a local replication of the virus, local inflammation, allowing then the spread of the virus and the systemic disease, which can be severe or not severe depending on the species. And you can see here that in susceptible bird species, there is a very prominent inflammatory response there, with also vasodilation, so vascular changes, and all of this probably explains how the virus is able to amplify there and then to spread systemically and cause the clinical signs that we observe with neurological conditions in these birds. Okay, so we decided to focus a bit on that. And what we know from the studies that are published on plenty of these orthoflaviviruses is that in the initial amplification of the virus, when it is inoculated by the mosquito, we know that dermal cells like dermal dendritic cells, but also dermal fibroblasts are very important in that initial amplification, and also the role of keratinocytes. And there seems to be an interplay between all these cells, because once the cell is infected, it's changing its expression of RNA and producing cytokines and chemokines, which will affect the permissivity, so the susceptibility, if you like, of other cell types to get infected and amplify the virus, but also impact the blood vessels. So we wanted to focus on that part. And there's another reason why we wanted to focus on that part, is that in resistant bird species, like chicken, for instance, there is no live virus which is going into the blood. So there is no viremia. Okay? Here, in these birds, you have strong viremia with a spread of the virus in different tissue. But here, actually, if you do the same injection with the same virus, it's stopped at the level of the skin. So it means that the initial response of the host at the skin level is probably what will determine the outcome of the infection. Okay? So we decided to focus mostly on that. What we know as well is this is immunohistochemistry in the skin of a bird which has been infected with UZO2 after 24 hours of infection, and one can see that these elongated cells here, which appear bright red positive with for UZO2 antigens, are dermal fibroblasts. So we know that these dermal fibroblasts are targeted by the virus, amplify the virus, and probably have an impact on surrounding cells. You also have other cell types like these round cells here, which are more histiocytic in nature, which are also probably important. And we also have a lymphoplasmacytic infiltrate here, which might be important for the pathogeny, but which doesn't seem to be infected by the virus itself. So this is quite interesting. So we need to decide how to go further. And the thing is that working with wild birds is ethically speaking not possible in Europe. So we had to use alternative models, and we decided to go for in vitro models. And we decided to study first the role of these dermal fibroblasts, and this is the focus of the PhD of Felipe, because there are plenty of papers on dengue, for instance, showing how important these dermal fibroblasts are in the initial response to the viral infection. So isolating cells from wild birds is not easy. We cannot catch them and sample them. It's not allowed in Europe. So what we had is to rely on birds which were found dead by people, bringing the cadavers of these birds. And isolating cells after the death of animals is very difficult. Primary cells are quite susceptible and quickly die. But there are cell types which survive for several days after the death of the animal or the person, whatever. These are the stem cells, mostly tendon-derived or mesenchymal stem cells, such as tendon-derived mesenchymal stem cells. And this is what we validated as a tool. This was our first study, isolating these cells from fresh cadavers and showing that actually when you compare these cells with the reference cell type which we are using for the amplification, well, this is globally the reference, the gold standard to amplify flaviviruses, Vero cells, you know probably these cells. And when we compare the permissivity of both cell types, actually it's not that bad. These are uninfected cells, and here you can see that both cell types, the reference and the mesenchymal stem cells from blackbird, can get infected by the virus, and the replication kinetics are not that bad. I mean by that, okay, here this is what we observe in Vero, this is the cells from blackbird, but this specific strain of the virus has been passaged 7 times in Vero cells, so it's highly adapted actually to Vero cells, but still it's capable to grow efficiently in blackbird cells. So we decided to use this model to try to compare what is going on in chicken and blackbird. So we isolated the same cell type, so mesenchymal stem cells from both species, and started to compare the replication kinetics. One can see here that using the same strain under the same condition, systematically we had more copies of the virus generated upon infection of the blackbird cells compared to the chicken cells, meaning that probably there's a better amplification of the virus there. The first thing we checked is it due to a difference in terms of the concentration or nature of the receptors to the virus at the surface of the cells? So we did what we call in virology a binding assay, and there is no significant difference there. So it's probably not related to the binding, the ability of the virus to bind to the cells. It's probably going on within the cells during the replication cycle. And one thing that might explain the difference is, of course, how the cells do respond to the infection. And to study that, we need to do what we call transcriptomic analysis. And so this is what we will do just after. And why is it important? Because these fibroblasts seems to be central in the initial response to flavivirus infection by expressing different chemokines, cytokines, which can trigger the arrival of incoming leukocytes, and they can also, of course, have a direct antiviral activity through the production of interferon, for instance. So this is why we wanted to compare this. So we did this transcriptomic analysis, what we call bulk transcriptomics. So we took a piece of tissue, extract total RNA, and then analyze the expression of messenger RNA. And what we observe is that there is a higher recruitment of permissive etiologic cells through the overexpression of chemokines in blackbirds, lower interferon response in the blackbird cells, and higher apoptotic response. So all in all, it means overall it means that there is a higher viral replication and dissemination of the virus in blackbirds. Okay, this was one way to address the question. Then we said, okay, it's interesting, but it might be that this virus is the result of the evolution of the virus coming from Africa, which adapted through evolution itself to specific bird species. What if we try to adapt a viral strain to chicken? And how this will change the pattern of expression and the behavior, the host pathogen interaction, if we do the chicken adaptation. So we decided to do so because we had shown that chicken embryos in eggs can get infected with the virus, not so efficiently as in blackbirds, for instance, but still we can manage to do that. So we decided to do serial passages of the virus in eggs. And if you do that, we did that with 2 different strains of the virus. You inject the virus, it's infecting the embryo, and then you collect the new virus and passage it to the next eggs and so on for a series of passages. What we observe is that by doing so, we can, from a very low mortality rate of embryo, go to a very high mortality rate of embryo. So definitely there is a selection for virulence in these chicken embryos. You can see here all the different mutations that were acquired during the adaptation process. And actually, which is surprising, is that we did it with 2 different strains of the virus from 2 different lineage. But there is no common mutation except some mutations here in the 3' UTR, which seems to be systematically present. Otherwise, there is no common pattern in the mutations we observe here. Okay. So the question is, by doing so, can we reproduce the blackbird response in chicken using a chicken-adapted virus? Okay. So once this had been done, we have the chicken-adapted virus, and we inoculated young chicks, very young chicks, just after the hatching, either with a parental virus adapted to blackbird or the new virus adapted to chicken, and compare their response by transcriptomics. What we did there is doing single-cell RNA sequencing. Probably you know about that a bit, or I can explain briefly. So the principle is that you can take some tissue or individual cells from the blood, like PBMC. But if you're working with tissue, you need to dissociate the tissue enzymatically usually. And when you have single-cell suspension, the machine will actually produce droplets. And there are different technologies used for single cell, but this is the most widely used technology. And in each droplet, actually, you have a single cell with all the reagents necessary for the transcriptomic analysis. Okay, so all the transcriptome you will get for each droplet is specific to one single cell. Okay, which is interesting because you can see what is going on specifically for all cell types which were present in the tissue. It's very different from the bulk analysis, where it's kind of a mix of a median expression value for all the cell types. Here it's very specific. Okay, this is how it's working. It's not important. What is very nice is that when you do the bioinformatic analysis of this single cell data, the computer will perform what we call the clustering. The clustering is actually grouping all the cells which have the similar transcriptome, which have the similar mRNA expression pattern. Some specific RNAs are highly expressed, others are not changed in terms of expression, and others are typically underexpressed in all these cells, and this is how the computer can group them together. Okay. So here actually these were very young chicks, so there were a lot of blastic cells. Those are actually cells which are going through the blood to colonize all the growing tissues to permit the maturation and evolution of these organs. So this is not probably something important for us. And these are all the leukocytes, the mononuclear leukocytes from the blood. So Mostly we have the lymphoid cells and myeloid cells. So if you focus on these cells and you reanalyze them, you have all these here with the lymphoblastic cells, and here the T and B cells, and here all the myeloid cells. Okay, when you compare this in the blood of these infected chicks, you can observe something very interesting. This is the control birds with all the lymphoid cells, myeloid cells, and this is the monocytic population. And you can see that there are not so many monocytes. But when they are infected, either by the chicken-adapted variant, or the parental variant, there are much more monocytes. But here, with the parental strain, there is a very specific monocytic population which is appearing. And when you dive a bit into the expression pattern here, you can see that these are highly antiviral, meaning that actually there is a strong antiviral response here against the parental virus, which is not present here because the virus has become able to control the host antiviral response, which is a sign of adaptation of the virus. So this is really interesting, these host-virus interactions. This is the expression pattern. This is called the heat map, where you can see all the overexpressed and underexpressed genes. So this is a code color for all the different clusters in the different conditions. Anyway, what is interesting is that mostly here for the adapted versus the parental strain, here you have plenty of overexpression of interferon-dependent antiviral genes, meaning that the host is able to control the viral infection. And here you do not have all these genes anymore, and you have the overexpressions even of some genes which are known to favor the replication of the virus. So it's very interesting to observe how this adaptation process of a virus to a new host species is making this virus able to better control all the cellular mechanisms which normally are able to control the virus. So this is what we observe, mostly the impact on the host interferon response. Okay. So this is a first example. And now let's discuss about the situation in mammals. So what we know in mammals is that there are, of course, differences between species. There are some highly susceptible species to orthoflavivirus infection, like dengue in people, Zika in people as well, West Nile in plenty of species, like in horses, but other species are quite resistant, so we do not know exactly why. But what we know as well is that there are very big differences in terms of susceptibility between individuals. Some people get infected by dengue. I have been infected with dengue, got nothing. My wife was infected with dengue, was severely sick with the infection during the same trip, and it happened. We do not know why. So there are reasons why. This is true, this is not a joke, it happened in Vietnam. And this is classical for these viruses, that there are really some individuals which are extremely susceptible to the infection, while others are completely resistant, and we do not know why. And this is a big problem. Dengue is about 400 million cases of infection per year globally. It's a huge problem. So, of course, we cannot take volunteers among the human population and infect them, so we had to work with models, and what we use mostly is working with mice. The thing is that mice are intrinsically quite resistant to flavivirus, so it's not so easy to use. Most studies have been using IFNAR negative, so interferon alpha receptor negative mice. These mice actually are not capable to respond to the interferon response. So, of course, they are highly susceptible, but they are susceptible to anything. I think it's not a very useful model in this case because we want to study the innate immune response. Okay, so this is not a good model for us. But immunocompetent mice are really extremely resistant to the infection by all these orthoflavi, dengue, Zika, all of them. We observe that sometimes when you infect adult immunocompetent mice, sometimes one individual, you don't know why, start to develop neuroinvasion and classical signs of encephalitis and strong replication of the virus here, strong inflammation, and eventually this specific mouse will die from the infection. So even if these mice have an homogeneous genetic background, they are congenic mice, they have all the same genetic background, so it's not a genetic reason. Well, it can be a genetic reason, but for other reason I will explain just afterwards. But with the same genetics, some will develop the disease and others not. So meaning that probably there are other factors that we do not know. They are still different from each other because, as you know, during the maturation of lymphocytes, the TCRs, the T cell receptors, and the B receptors, the repertoire of these receptors is individual specific. It's a haphazard event occurring during the maturation of lymphoid organs. So for the same genetics, you can still have a different adaptive immune response. But the thing is that is happening quite early after the infection. So it's probably more related to the innate than to adaptive immune response. So there are still a lot of unsolved questions behind this. We also wanted to address the impact of age in mice. Why? Because we know that Zika virus, you know, is highly problematic in newborns and children that have been infected in utero. And this is where you have the most devastating lesions. Dengue is more severe in children. This is well known. Japanese encephalitis also. So definitely there seems to be much more important susceptibility of young individuals to many orthoflaviviruses. So what about UZU2 in mice? And why do we also have all these models in the lab, but we like UZU2 virus, and there is a very practical reason for that, is that UZU2 virus is the only member of this genus that can be handled under BSL-2 conditions, so biosafety level 2 conditions. All these guys require BSL-3 conditions. We have a BSL-3 which is extremely busy with COVID-19 products and H5N1 influenza products. So we try to spare this lab and focus on UZU2. And what we do actually is what we observe with this model of orthoflavivirus. Then we can confirm it with other viruses in the BSL-3. Okay, so this is why I like this model a lot, but it's a quite interesting virus anyway. And this is very, very nice because you can see here, this is the survival rate of young mice after infection. You can see that mice from 2 weeks of age all survive the infection, like do almost all the adult mice that we infect. But younger mice all die from the infection. This is really on a very short, narrow window of time, a complete change of the phenotype from high susceptibility to high resistance. This is quite exciting. It means that probably for babies it could be quite dangerous to get infected by such viruses. And we can also see, you see the tropism for the virus here for the brain compared to liver and spleen, which are two important organs for the spread of the virus. Liver and spleen are heavily infected in bird species, for instance, and you know from the pathogenesis, from yellow fever, dengue, and so on, that there is a strong replication in the liver. But there is really a neurotropism of this virus. It's really a neurotropic virus like West Nile. And you can see the lesions in the brain with gliosis, with vasculitis, and plenty of degenerative changes in the neurons. Strong replication of the virus, okay, in plenty of places in the brain and the spinal cord. So this is what we observe, this big shift from 100% mortality to 100% survival in a matter of days of maturation. So we decided to focus on this question because this is something very exciting and interesting. To try to bring, to address this question and bring some pieces of information, we compared the kinetics of replication of the virus at different time points after the infection in the skin. And we use footpad infection in these mice. And we can see that at the level of the skin itself, so when you inoculate the virus in the footpad, there is not significant differences between the susceptible and non-susceptible mice. So it's probably not there that the things happen. Well, not at least in terms of pure replication of the virus. So the virus is able to replicate in the skin of young and slightly older mice. But in the blood, in the liver, and mostly in the brain, there you have huge differences. Okay. Another important question is that if you read a textbook on orthoflaviviruses like dengue or Zika, whatever, all the books say the virus first replicates in the skin, then there are some leukocytes which get infected and used as Trojan horse to allow the virus to spread, to get access to the brain, for instance, and so on. So this is what we call the cell-associated viremia. And our question was, is Usutu virus spreading through cell-associated viremia or alone as a virus, cell-free virus into the plasma? And it appears that there is no cell-associated viremia for Usutu virus. And actually, when you look a bit deeper into the literature, most information we have tend to show that this is not so important, actually. Most of the virus actually is free in the plasma, probably from the skin, and then either through the lymphatic system or directly through the blood, going into the bloodstream and then spreading to other tissues. And so we wanted to assess a bit what was going on in terms of viremia. And what we observe, interestingly, is that actually all young mice infected by the virus showed high amounts of infectious virus, while in the 15-day-old mice, most of them had only viral RNA, but no infectious virus. Meaning that, again, a bit like we observed in the birds, the skin barrier seems to be key in the outcome of infection. Meaning that probably in the 15-day-old mice, which are resistant, the virus replicates in the skin, but still remains confined and limited to the skin. It's not able to spread beyond the skin, while in 9-day-old mice, the virus replicates in the skin and then is able to spread to the brain and then induce the disease that we observe. Okay. So this is what is shown here, this systemic spread of the virus from the skin to the brain, possibly directly or through key organs like the spleen or the liver, while it is blocked at the level of the skin barrier in these older mice. Okay. Again, since the skin barrier seems to be determinant for the outcome of infection, we wanted to confirm that. If this hypothesis is true, it means that if we bypass the skin barrier while injecting the virus intravenously in these mice, they should die. Okay? So this is what we decided to do. So one way to inject is, these are very tiny mice, so injecting into the skin, to the tail veins is extremely difficult. So we are using a blood sinus which is just behind the eye. It's a very easy procedure. And when we do so, so this is the initial mortality rate from the skin infected mice. So here all the 15-day-old mice survive and all the 9-day-old mice die when we inject them in the skin. What happens if we inject the virus intravenously? We can see the shift in mortality from 100% survival to almost 100% mortality here in these 15-day-old mice. It means that indeed the skin barrier is really a very important determinant of the outcome in these young mice. Based on that, we wanted to dive a bit further and deeper into understanding what was going on there and comparing the two phenotypes. So the first thing we did is to address by proteomic analysis the expression pattern of some key chemokines and cytokines in the blood of these mice. And one can see that here, so in blue, it's the 9-day-old mice, and yellow and red are the 15-day-old mice. For some specific chemokines like CCL2, for instance, you can see the quite big difference in terms of expression. Why is it important? It's because it's an attractant of some key cells like neutrophils and some monocytic populations. The same for interleukin-1. Interleukin-1 is very important for the vasoactive response. So it's a key cytokine from what we call the cytokine storm. known for SARS-CoV-2, for instance. So it has very strong impact on the blood vessels, the activation of the blood vessel, the permeability of blood vessels as well, and the attraction of incoming leukocytes. Also plenty of others like interleukins and so on. So there are some very interesting differences here, but the most interesting ones are these chemokines and interleukin 1. A bit like we did with the birds previously, we did single-cell RNA sequencing in the skin of all these mice, and this will be the last part of the study I present you. And we wanted to compare what was going on there. So using this single cell, what we do, we took skin pieces at the place where we inoculated the virus, and we also had control mice, uninfected mice. We dissociated the tissue and analyzed the transcriptomic pattern of expression of all these cell population, and you can see here the image of the clustering. So you have fibroblastic populations, all the keratinocytes here, from the basal cells to the more keratinized cells. You have some, these are young mice, so some erythroid cells as well. You have the endothelial cells, and here you have mostly mast cells, B cells, and myeloid cells which are present. This is another graphical representation of all these populations across the different samples. So these are the 15-day-old mice, uninfected and infected, and this is the 9-day-old mice, uninfected and infected. You can see that here the different clusters of cells are quite similar in all conditions, but you can see that there is a big shift here with some specific clusters are appearing. It's a quite different image in terms of clustering when you compare these three and the last one here. So this is something interesting. It means that upon infection, the 9-day-old mice respond differently from all other conditions. When you zoom on the myeloid cells, so myeloids are all the monocytic, histiocytic, and neutrophilic, so granulocyte cells. You can see that 9-day-old mice, 1 day post-infection, compared to 15-day-old mice, 1 day post-infection, they have a different profile. Here, regarding the lymphocytic cells, you have a specific population here, which is not present there, or almost not present. And regarding myeloid cells, you have this population and this one, which is very interesting, and these are neutrophils. So neutrophils are present here and not here. So the response here is completely different. Why is it important? Because neutrophils, for instance, do produce a lot of molecules which will affect the permeability of blood and lymphatic vessels. And our hypothesis is that probably this attraction, the release of chemokines like CCL2, for instance, which is attracting neutrophils, is making the skin more permeable to the virus, allowing the virus to spread either directly to the blood or to the lymphatics and then to the blood to other tissues. So this is a quite interesting image we have here. And when you look in terms of the pattern of expression, you can find exactly the same molecules that we highlighted in the blood by the proteomic analysis. Here, CXCL10 is overexpressed in 9-day-old infected mice. CCL2 is overexpressed in the same mice and not in the others. Interleukin 1, similarly overexpressed specifically there. But you also have some very interesting molecules which are extremely expressed here and almost not in the other conditions, like matrix metalloproteinase 9 and this other factor here. And these two factors, especially that one, are extremely important. They are really well-known factors involved in crossing, for instance, the blood-brain barrier by flaviviruses, like West Nile, for instance. If you have MMP9 KO, so knockout mice, they are resistant to neuroinvasion by West Nile. So the blood-brain barrier is an endothelial barrier, the same as the skin endothelial barrier. So our hypothesis is that this overexpression of matrix metalloproteinase is actually destabilizing the endothelial barrier of the skin and allowing the virus to spread through and get to the bloodstream. Okay, so now we are using different strategies, using microRNAs to prevent this expression locally in the skin in the mice, to study how it can make the 9-day-old mice more resistant to infection. The advantage is that for many of these molecules as well, there are chemical inhibitors which are validated, like for this one, for instance. So we have plenty of strategies going on now to study that question. And this is also an interesting image. This is a dot plot showing the different conditions: 15-day-old mice, uninfected and infected; 9-day-old mice, uninfected, infected. And you can see that there's also another important factor here which is very specific to that condition. So the 9-day-old mice infected, which is called Schlaffen 4. It's a German name. And this Schlaffen factor is actually a proviral factor. It has been shown to promote viral replication. And again, there was a very nice study with West Nile showing that if you knock down this factor, West Nile virus is not able to replicate anymore. So the overexpression of this factor here is probably something which allows a better replication of Usutu virus in these young individuals. Which is interesting is that you know that young mammals have a biased immune response towards the Th2 response, which is an allergic-like reaction. And we actually have plenty of evidence now that in adult vertebrates and mammals, allergic individuals are more susceptible to flavivirus infection, probably because they have a different reaction to the mosquito bite, allowing a more important inflammation there, a more important vasodilation, and probably an easier way for the virus to spread systemically. And this is why we are trying to understand really the mechanics there, what is underlying this susceptibility of young animals, because we believe that this might, at least in part, explain the individual specific response in people when infected by flaviviruses. And I think we are arriving to the end of this presentation. So what we observe all in all in these young mice is that there is an increase on the T-cell permeability and higher viral replication, and combined together, this explains the specific phenotype of these mice which die from the infection, while slightly all the mice survive the infection. And I will thank you for your attention and would enjoy answer your questions. Sebastian.
Well, thank you for your outstanding presentation. I'm thinking on birds, on wild birds as carriers of many viruses. And you were talking about the Usutu virus, and for Usutu virus, it seems that passerine birds are very important carriers. So, are there any study on passerine birds as host of Usutu virus in South America? And the second question is, would it be efficient or effective to collect blood with some DNA stabilizers, such as RNA-later or RNA shield, to see if we can find the virus? Because I think it will be easier to collect small amounts of blood rather than collect a big sample and to obtain serum and search for antibodies or something.
Actually, this is something I wanted to discuss with you.
Okay.
Because I think birds are the key. As I show you this image, the reservoir of most of these viruses are birds. And so collecting the blood of birds is very important for the surveillance. It's much more sensitive than sampling people, for instance. So I totally agree with you. Not only to look for the virus itself, but because you need to be lucky, and at the time you sample the bird, it must be infected just before, be during the viremia step, which is a very short, narrow window of time. So yeah. So what we prefer to do is to do serology anyway. But actually we developed some micro testings in ELISA and serum neutralization, allowing us to do some good serology from 5 microliters of sample, which is very few. Yeah. And why did it validate? Thomas knows about that story, but because we have a project in Indonesia where actually we use mosquitoes as blood samplers. So we collect blood-filled mosquitoes, which usually have in their abdomen about 5 microliters of the blood when they fed on people or animals. We characterize this blood to tell, okay, which species did this mosquito feed on. And because there we focus on macaques in this area, and we are sure this is from the right species, then using these 5 microliters, we can do a couple of serological analyses for flaviviruses and alpha viruses like chikungunya and Western, well, all the equine encephalitis virus. So that would be the best way to proceed, to have a very quick view of the circulating viruses. Then, obviously, it would be interesting also to have this phylogenetic analysis once you have confirmation of the circulation of some virus in some area. Like this Uzutu strain in Colombia, to me it's quite interesting to know which strain of the virus it is, because the clinical presentation is people has never been described before. It's more like yellow fever. It doesn't sound like West Nile. So it's not encephalitis, it's more like a liver and systemic disease. But it's quite severe and yeah, it's unusual. So is it a new strain from Africa which emerged there in Colombia, but then how did it appear in Colombia? Is it already present elsewhere in South America? These are important questions to me. Yeah.
I have
One more, just in the same way that we were discussing. So for that you will need a fresh sample of blood, or?
No, frozen.
Okay,
It's fine.
Frozen?
Yep, minus 20. I think we can discuss something.
Okay.
I'm sure.
Would the thickness of the skin be an important factor, or is similar?
Sorry,
the thickness of the skin of these animals, or is?
That's a good question. Not that I know, but there is no really studies addressing that question. I don't think, because I think that examples of severe disease associated with flavivirus has been described plenty of vertebrates, regardless the thickness of the skin. And you know that, for instance, West Nile is described in reptiles, severe West Nile causing lethality. They have a very thick skin, but actually you have some mosquito species which have a tropism for some specific vertebrates like reptiles, for instance. So I don't think so.
And the last one, if we follow your experiments of the domestic chicken eventually could be infected by this virus and become a reservoir?
Yes. The thing is that it's an evolving process. So viral evolution, well, like the evolution of all organisms, is a dynamic process. And depending on the opportunities for cross-species transmission, it may occur at some time that it becomes a problem for chicken as well. Yeah.
More questions?
I have one.
Yep.
This is a very common, because I'm not related to the field, but now I'm not worried about the infection. So this is I mean, all the mosquitoes can transmit the infection, or there is a specific one, or there is an environmental condition that can favor the transmission? Or?
Of course, this is a very complex question, because, for instance, in Europe, the main mosquito species involved is Culex pipiens, but actually you have different varieties of these Culex pipiens mosquitoes, and all the varieties do not have the same feeding behavior. Some are ornithophilic, they feed on birds, and some are anthropophilic, feeding on people. And so sometimes creating the bridge between birds and people is not that easy, ecologically speaking, because mosquitoes have a really specific tropism. So it's sometimes a matter of chance, if I may speak so, that a virus spread to another species. And on the contrary, there are some other mosquito species which have a very broad host spectrum. And then, of course, these guys are much more dangerous. And again, this is something which is evolving over time. So actually, when a virus is present for a long period of time in a given place, actually it can adapt itself to local mosquito species, local bird or other species of reservoirs, and potentially to get transmitted to people. So what is true right now in a specific continent or geographic area might not be true elsewhere or maybe later in the future. So that's why we need to have this continuous monitoring. But, well, to survey everything all the time is useless. But when something weird is appearing, like these cases in Colombia, I think it's worth checking and seeing what we can do. Yeah.
Thank you.
Okay, any other questions? Well,
I'm sorry. I have a general question because the wildlife science and virus dynamics is not my area, but in a research in veterinary science, maybe it's a philosophical question, but what's in your expertise, Professor Mitian? What is your cornerstone piece in starting a research in all the areas, maybe public health and wildlife science or something? What is the motto or objective? Maybe a question, a correct question in all these researches.
That we are performing, I mean.
Yeah.
It's complex. At the beginning, I was really interested in these viruses because of the outbreaks in wildlife, and actually there was a very impressive drop in the population of many bird populations, like house sparrow, for instance. There was a 90% drop in the population, and there were plenty of suggested culprits, but it seems that these viruses were involved in the drop in population. What is interesting is that over a very short time, after a couple of years, there was a strong population immunity which appeared in bird populations, and they have become quite resistant now to the infection. So now we are in a situation of endemicity where actually sometimes some birds get sick and die, but most of the birds actually are quite resistant because of this population immunity. And it's quite usual actually after an emergence, you have this very important impact, and then it's probably part of the landscape, I would say, ecological landscape. And there are this also, what is also motivating me is to all these interactions between viruses. Not seeing viruses just as a cause of disease, but as rather organisms which evolve and try to get adapted to the new ecological niche. And they are competing with each other, and this is quite interesting because this is an opportunity in the field of vaccinology, for instance. We do not have vaccines. Even the vaccines against dengue, they are not so good, they're not so efficient. They can be even dangerous because they can favor the production of non-protecting antibodies, which can actually worsen the clinical evolution of patients, which is called antibody-dependent enhancement. And this is the main limitation in developing vaccines against these viruses. It has been described for COVID-19 as well. So all what we observe is very interesting. What I did not present to you is but that would be interesting maybe someday that I show you that, but what our Italian colleagues are doing is that, and it has been shown in people as well, many people which were infected by Usutu virus, and it's the same for mice, do not develop detecting or detectable amounts of antibodies. So many remain seronegative. This is the limitation of serology, at least in mammals. But they are protected anyway against West Nile challenge. So it means that the cellular immune response is very important. And there it's a broad field of study because most of the vaccines we are usually using in public health are based on the development of antibodies. And this is usually the outcome which is sought when you want to validate a new vaccine, just checking, okay, there are a lot of neutralizing antibodies, I'm happy. But actually, no, the cellular immune response is probably even more important. So we need to develop new tools so you know that there are some vaccines when you can use specific adjuvants to orientate the response toward the cell-specific response, so CD8 T cells. It's working actually. So that also an interesting link between the observation of fundamental science and vaccinology, because the main, well, the best response to that is vaccination, of course.
Yeah. Thank you.
Okay.
My
Thank
pleasure.
you very much. I want to acknowledge Thomas, which I didn't mention at the beginning because he was the person who brought Museum and they had this collaboration. And I want also to mention that among us, we also have undergraduate students, which is very, very nice to see you here, and I hope that you will be the next PhD or Magister student. I thank you, Museum, for your kindness and the fantastic talk. And I wish you a very successful academic year because we are starting an academic year, and now we invite you for a non-alcoholic cocktail over there. Thank you very much.